All right, so here we have the cell cycle. It consists of three major stages. We have interphase as the first stage, mitosis as the second stage, and cytokinesis as the third. So if you look at your note guideline, you're going to see the cell cycle, 1, 2, 3, that's where you're filling that out. So just make sure you're filling it in. So looking to the right here at the pie chart, all you math people should be able to tell me, which of these three stages is a cell cycle in for the longest period of time? So is it interphase, is it mitosis, or cytokinesis? What do we think? Julia? Yes, good. So here we have interphase. If we look at the pie chart, this whole section is interphase. So this entire time, the cell is in that stage. This piece is where the actual cell division takes place. So during this time, no cells are dividing yet. It's just getting prepared for it. Then we have mitosis take place, then finally cytokinesis, and that's the ending of the cell cycle, where the cell will have divided and become two separate cells. When we look at interphase more carefully, we're going to see why it takes the longest period of time. So remember, blue is the color for us writing it down. In interphase, two major things are happening. Cells growing to mature size, and it's making a copy of the DNA. Those are the two major pieces.
How do you get a copy of that sheet?
So after that DNA is copied, how many sets of DNA does a cell contain? We started with one, then it makes a copy, so now we have a total of what, everybody? Two. All right. So this could last about 22 hours. So that's what we mean. It's taking a long time for the cell to grow to mature size and that DNA be copied. But that's a really important thing to kind of set the stage for the rest of the cell cycle. So next up is our stage mitosis. And within mitosis, there are 4 phases that we're going to learn about. So you're going to see a lot of organization take place here. And this is where the actual cell division starts to happen. So remember, in interphase, it's just preparing for that. This is where we start to see division take place. And remember, from our cell theory, new cells are being created from old cells. So that cell's nucleus, in addition to the other organelles, also have to divide up so that at the end we have 2 new nuclei. One set of the DNA is going to stay in one nucleus, and the other set goes into the other nucleus. So let's look at these phases more specifically. This is kind of the overall view of what would happen in mitosis. So if we look at the top here, you're going to notice that there's one cell with some DNA inside. Okay, so we can imagine that this is towards the end of interphase, where the DNA has been copied. So let's say we just started with the 1, and now we've got 2 in there. Once interphase is complete, we're now ready for mitosis, where we can start the different phases. So you're going to be introduced to a phase called prophase, and during prophase, those chromosomes now get condensed into these little X's, and they all come together in the center. So in metaphase, it lines up along the middle. Anaphase is where those little spindle fibers we talked about before that were connected to the centers get split apart. So now they get pulled to opposite ends of the cell. Okay? So we're almost there where we have 2 cells of the same DNA, but not quite yet. We need that final split to take place, which is that last stage of cytokinesis. So you should notice that the very first cell up here, right, the original cell, looks identical to the 2 ending cells. I don't know what happened there. Okay? So they each have the same DNA inside of them at the end that the original one started with. So let's look at these phases more specifically. in prophase. Okay? This is when that chromatin, which is basically the word for chromosome before it's into these nice, neat X's, in the nucleus is going to condense together and form chromosomes. So now we have all these X's in the center. The other thing that's happening at this time is those spindle fibers that we looked at before are starting to form. Here they are, these little guys. Okay? They're going to play a really important role later. Right now, they're just forming. We all good to move on? So the next phase that's going to come up is metaphase. So you want to take note of what's going to happen to those X's when we enter the next phase. They're right now, they're just clumped together. And when we go to metaphase, we're going to see those X's now neatly lined up across the center of the cell. You also see that those spindle fibers that were forming before now have attached themselves to the center of each of these X's. Okay, each of the chromosomes. And that's for a very important reason for the next phase coming up. So in the next phase, in anaphase, you're going to see that those spindle fibers do what? Does anybody want to take a guess? If they're attaching to the centers there, and our ultimate goal is to end up with the same DNA in each cell, what could happen now to those X's? They're going to do what? Those spindle fibers are attached to the center so that they can be divided. Good. Pulled apart. Divided up. So this is what it would look like in anaphase. So those chromatids separate and they move to opposite ends of the cell. So we're almost there. We almost have two cells with the same DNA, but they're all still stuck in the same cell at this moment in time. So we need a little bit of adjusting to do. And that's where our next phase comes into play, and that's telophase. And in telophase, you're going to see that now that they're at opposite ends of the cell, we can start to see new nuclear membranes form. All right, in telophase, new nuclear membrane forms around each group of chromosomes. So you can see here that there's a little nuclear envelope forming around each new set. And we're getting close, right? We're kind of starting to see the cell pinch into two. We're not quite there yet, but we're getting almost finished to the final product of two identical cells with the same DNA. Cytokinesis, the final stage of the cell cycle. We have the cell finally pinching into two. So each of those daughter cells, as they call them, are going to end up with the same number of identical chromosomes. So our final product is the same as what the original cell looked like, but now there's two.